Okay, so in Chapter 11, Part 2, we're going to finish up our talk on the process of meiosis. And so remember that we left off in the previous video with telophase 1 and cytokinesis. And so now we're going to enter into meiosis 2, the second round of division. And just to make sure that you understand this, so we went from one diploid parent cell to two haploid daughter cells by the end of meiosis 1. And so by the end of meiosis 1, again, we have two cells, and both of those cells are haploid, meaning that their genetic information has been reduced by half. Okay, so in humans, say for example, the cell that we would start off with would have 46 chromosomes, and by the end of meiosis 1, we'd have two cells, and each of those cells would have 23 chromosomes. The cells' chromosomes would be in the dyad form, but there would only be 23, so half of that original number. Okay, and so now we're going to enter into meiosis 2. So we have two cells that are going to go through this second stage. And let me just move my camera. So in some species, there's a phase in between meiosis 1 and meiosis 2, and it's called interkinesis. And so I put that on here. Interkinesis can be thought of as a gap, and it's a gap that can be very long or very short, depending again on the species. So it's a gap between meiosis 1 and 2, and the key thing I want you to remember here is, the cells would be, alive and doing things, normal metabolism, but DNA replication does not happen again here. That's a key thing I want you to understand. The only time DNA replication occurs is in interphase, which precedes meiosis 1. Okay, so after interkinesis, if it happens, the cells, both cells, will enter into prophase 2. And so in this picture here, I'm showing you two cells, and both are entering into prophase 2. So what happens here? So if the chromosomes decondensed at all during interkinesis, the chromosomes will recondense. So they'll recoil back up into those structures that we call chromosomes. And they are dyad chromosomes, so two arms. The nuclear envelope will break down again. The meiotic spindle will form, and so you can see here, these are animal cells, so there's centrioles at the heart of the meiotic spindle. Okay, same thing over here. Here's another set of centrioles, and there's that meiotic spindle forming. So in this nucleus, I have 1, 2, 3 dyad chromosomes, and you can see that there was some crossover that happened, right? In this other cell's nucleus, again, I have 1, 2, 3 chromosomes. This chromosome here and this chromosome here were a homologous pair. This chromosome and this chromosome, this chromosome and this chromosome, they were 3 homologous pairs back in meiosis 1. Those have now ended up in 2 separate cells. Okay, so again, I've got 3 chromosomes in each cell. No homologous pairs are here. And again, at the end of prophase 2, the nucleus will be completely broken down. Chromosomes will be completely condensed, and that meiotic spindle will be set up. We then enter into metaphase 2. So the phase names are the same, right? Nice and easy. So when you think metaphase, I want you to think middle. And so during metaphase 2, what will happen is the meiotic spindle will attach to each dyad chromosome, and each of those dyad chromosomes will line up single file now in the center of the cell. So I just gave you 2 examples. And so here's these 2 cells, okay? Or we can also look at these 2 cells. This is what we saw on the previous page. And so again, chromosomes are lined up here in the middle. There's 3 here, and there's 3 here, just like we saw in the previous slide, 3 in those nuclei. These cells up here only have 2 chromosomes each. So this is a different species. Okay, so next comes anaphase 2. And so anaphase away. So here we're going to have the arms of the dyad chromosome are going to be separated from each other. So the proteins that are holding those chromosomes together, they will break down, and the sister chromatids will be pulled towards the poles. Okay, so again, we'll come to these cells. These were just 3 dyad chromosomes in the middle during metaphase 2, and now the arms of those dyad chromosomes are going to be pulled in opposite directions, so we say to opposite poles. Okay, and the same thing is happening in this other cell. Next comes telophase 2, and so telophase 2, those monad chromosomes are completely pulled to the poles, right? Completely separated from each other. And the nuclear envelope starts to reform around the DNA that's been separated, and that DNA will start to decondense. Okay, so we can see here in this figure, this was originally 1 cell, and now you can see we have 2 nuclei forming. And the same thing here, this was originally 2 cells at the end of meiosis 1, and now we have 2 nuclei. Following telophase 2, we will go into cytokinesis 2, where the rest of the cell is divided. And so by the end of cytokinesis, you're going to have 2 cells here and 2 cells here. So we end up with 4 daughter cells at the end of meiosis 2. And if we were to look at the genetic contents of each of those 4 cells, we would say that they are haploid. They contain half the number of chromosomes that were present in the original parent cell. So again, for humans, that would be 23. So there'd be 46 chromosomes in the original parent cell and only 23 chromosomes in each gamete, the product of meiosis. Okay, and here's just another figure showing you those other cells. So I wanted to walk you through with both examples. And so again, this is going to be telophase 2, right? Chromosomes monad form, end up at different poles or different sides. The nuclear envelope reforms. And then in the end, cytokinesis, we end up with 4 individual cells. And again, all of these will be haploid. Okay. So one of the things that I'm going to ask you to do, that you should be able to do, is to compare and contrast meiosis 1 and meiosis 2. Okay, so this figure shows the major events of meiosis 1 and the major events of meiosis 2. So if we look first at the top here, if we think about prophase and metaphase, so kind of that pro-metaphase region, what we have in meiosis 1 is homologous chromosomes that pair up to form tetrads. And we have non-sister chromatid arms that could cross over, forming a chiasmata. And we could have genetic exchange. Okay? And so if we look at anaphase 1, we actually show some recombination, some genetic exchange. And we show that dyad chromosomes with sister chromatids stay attached to each other during anaphase 1. And so dyads are pulled towards each pole. So in anaphase 1, homologous chromosomes are separated from each other. In meiosis 2, we don't have homologous chromosomes. There's no tetrads. There's no crossover. Instead, single dyad chromosomes with their 2 sister chromatids line up, right, during metaphase. And it's these 2 arms that get separated during anaphase 2. So by the end here, we end up with cells that have monad chromosomes in their nuclei. Okay, so you should be able to compare and contrast meiosis 1 and 2. Here's a summary slide, just showing all the stages that we've talked about now from the previous video and this one. So again, you should understand what happens actually before meiosis even starts. So you should know what happens during interphase, right? DNA gets replicated. We go from 1 complete copy of the genome to 2. Prophase 1, DNA condenses and we form tetrads, right? Homologous chromosomes pair up, and this is where crossing over can occur in recombination. Metaphase 1, tetrads line up on the metaphase plate. Anaphase 1 dyads are pulled apart. Telophase 1 in cytokinesis, we end up with 2 cells. They are haploid, and chromosomes are in the dyad form. Prophase 2, DNA recondenses, nuclear envelope breaks down. Again, we have haploid cells, and chromosomes are in the dyad form. Metaphase 2, those dyad chromosomes line up on the metaphase plate in the middle of the cell. Anaphase 2, dyad chromosomes are separated and monad chromosomes are pulled towards the poles. In telophase 2, we end up with 4 cells. All of them are haploid. They contain half the genetic information as the original parent. Okay, so again, we go from 1 diploid cell to 4 haploid cells. And remember, they're haploid from the end of meiosis 1. Okay. Lastly, I gave you a summary of meiosis 1 and 2, and I'm not going to read this to you. This is right from our textbook, so you can look that up. So hopefully you can go through here and you can see what happens in each stage. So again, I kind of just did this on the previous slide, but you should be able to, talk about interphase, realize the important stage there is S phase, look at the outcome. So this is a nice summary slide of both meiosis 1 and meiosis 2. And on the last slide, we have a comparison. So this particular table compares mitosis with just meiosis, not splitting it up into 1 and 2, just generally meiosis. And so you can see both processes. So we can see meiosis, meiosis 1, meiosis 2, and the result here. And then we can see mitosis, which is just one round of cell division. So there's no 1 and 2 when we talk about mitosis. And again, we can see the end result. You can see that meiosis 2 and mitosis look very similar. Okay, it's meiosis 1 that's the big difference here. And so down here, I like this table. It shows you the process and it shows you the similarities or differences between meiosis and mitosis. So if we look at DNA synthesis, for example, in both meiosis and mitosis, this process occurs during S phase of interphase. So that's a similarity. So again, if I ask you to compare and contrast mitosis and meiosis, you should be able to do that. Second process, synapsis of homologous chromosomes. So when do those chromosomes come together to form tetrads? Well, let me jump to the bottom here. In mitosis, that never occurs because it doesn't happen there, right? There are no tetrads. Homologous chromosomes don't pair up. For meiosis, it's going to happen during prophase 1 of meiosis 1. Crossover happens when tetrads are formed. So for meiosis, that'll be during prophase 1. Doesn't happen in mitosis. Homologous chromosomes line up on the metaphase plate. For meiosis, it's during metaphase 1. It doesn't happen in mitosis. Sister chromatids line up on the metaphase plate. So not homologous chromosomes, but sister chromatids. That's going to happen during metaphase 2 of meiosis. In mitosis, it happens during metaphase. There is no 1 or 2, it's just metaphase. And lastly, the outcome. So the number and genetic composition of daughter cells. So in meiosis, we end up with 4 cells, and those 4 cells are all haploid. For mitosis, we end up with just 2 cells, and those 2 cells will be diploid. Okay, so again, you should be able to compare and contrast these two processes, and that is the end of the meiosis chapter.